Let me set the stage. Maize, or corn as you probably call it, is one of the world's top 3 staple grains, right up there with wheat and rice. Every year, global maize production tops a staggering 1.2 billion tons. But here's the kicker. As our population grows and arable land shrinks, we've got to make every drop of water and every grain of fertilizer count. You might ask, why focus on water and nitrogen? Well, water use efficiency, WU, and nitrogen use efficiency, NU, are like the gas mileage and horsepower of crop production. If we can boost these, we not only grow more with less, but we also limit environmental damage from excess fertilizer and water use. Now, here's the conflict. Maize is notorious for its low NU wages, 29% in developing countries. That means 2/3 of the nitrogen we add to fields goes to waste. It leaches into water, becomes pollution, and costs farmers money. But what if we could select or breed maize plants that thrive with less? Imagine a field where every plant sips water wisely and gobbles up nitrogen before it's lost. That's the goal, and it's where genetics steps into the spotlight. So how do we untangle the genetic roots of efficiency in maize? Spoiler, it takes more than a green thumb. It takes deep analysis of how genes control these complex, interconnected traits. All right, let's break down what we mean by water and nitrogen efficiency in maize, because these are not just buzzwords. First up, water use efficiency, or WUE. This is the grain yield a plant produces per unit of water it receives. Think of it as bushels per bucket. Then there's nitrogen use efficiency, NU, which measures how much grain you get per unit of nitrogen available in the soil. But wait, it gets more granular. NU actually splits into nitrogen uptake efficiency, NUP how much nitrogen the plant absorbs from the soil, and nitrogen utilization efficiency, NUT how well it converts that nitrogen into grain. It's like the difference between how much you eat and how much energy you get from your food. Now you may wonder, aren't these just simple traits? Not at all. Both WE and NUE are classic complex phenotypic traits, meaning they're shaped by a network of genes and influenced by the environment. Picture a mixing board with dozens of sliders, each representing a gene or an environmental factor. To complicate things further, improving one trait sometimes affects another, not always for the better. For instance, cranking up NU can sometimes lower WE, depending on the plant's physiology and external conditions. This web of interactions is exactly why plant breeders need solid tools and clever strategies to pinpoint which plants are truly efficient and why. Let's dive into one of the most powerful techniques breeders use: generation mean analysis. Imagine you're a detective, tracking down which GeneSor combinations of genes are behind a trait. Generation mean analysis lets us compare parents, their hybrids, and subsequent generations to decode the genetic action at play. Here's the setup. Researchers cross 2 inbred parent maize lines, then grow out the phone hybrid, the F2 generation, and back crosses. For each, they measure traits like WE and NU. The conflict? Each generation reveals a different story about how genes interact, sometimes additively, sometimes with dominance, and sometimes through epistasis. So, what are additive and dominance effects? Additive effects are like stacking bricks. Each gene adds a little to the trait. Dominance means one gene overshadows another, like an on-off switch. Epistasis? That's when gene pairs interact, sometimes amplifying or masking each other's effects. Think of it as a team sport, not a solo race. Generation mean analysis uses statistical models to estimate these effects, telling us whether selecting for a trait will be straightforward or a genetic roller coaster. If you're wondering, does this really make a difference in the field? Absolutely. Knowing the genetic architecture helps breeders predict which crosses will yield the best, most efficient maize plants. Now, let's unpack the real-world findings from a study of 3 maize hybrids in Syria. The researchers found something fascinating. For most efficiency traits, new grain yield dominance gene action was more important than additive effects. In plain English, that means certain gene versions can overpower others, creating big jumps in performance. But here's the twist: epistasis also played a major role. That's when the effect of one gene depends on others, like a jazz band riffing together instead of playing solo. This kind of gene interaction, especially duplicate epistasis, can actually make breeding more complicated. Now, you might be thinking, if dominance and epistasis rule, does that mean selection is hopeless? Not at all. The study also measured heritability, a technical term describing how much of trait variation is genetic versus environmental. High heritability, especially narrow-sense heritability, means you can make real genetic progress through selection. For traits like nitrogen use efficiency and biological yield, narrow-sense heritability was high. Think of it as having a reliable GPS for breeders. For water use efficiency, it was lower, meaning environmental factors like rainfall and soil type had a bigger say. So, the resolution? Breeding for efficiency is possible, but you need to know which traits are driven by which genetic mechanisms to pick your best strategy. So, how do breeders turn these genetic insights into better maize varieties? Let's walk through it. If dominance and epistasis are key players, you don't just pick the best plant and run. You need to create and test hybrids, sometimes across several generations, to capture the right gene combinations. For traits with high narrow-sense heritability like nitrogen use efficiency, simple selection in early generations can work wonders. Picture a coach spotting talented rookies and moving them straight to varsity. But for traits with lower heritability or tangled gene interactions like water use efficiency, breeders often use recurrent selection, cycling through several rounds of selection and crossing to gradually build up the winning combination of genes. Now, let's not forget environmental influence, a technical term called phenotypic coefficient of variance, PCV. If PCV is much higher than the genotypic coefficient, GCV, it's a sign that environment is muddying the genetic waters. Breeders need to test plants in different places and years to spot the true winners. Bottom line, there's no one-size-fits-all. Breeding for efficiency means tailoring your approach to the genetic and environmental realities of each trait. Let's bring it home. Why does all this